Today we will discuss about immunoglobulin G. It is most important antibody in our body. So in detail of this immunoglobulin G. So first about the structure. Basically this immunoglobulin G is made up of two heavy chain and two light chain, and they are combined to the disulfide bridges and making a quaternary structure of immunoglobulin G, and that is Y shape. Further, in case of this immunoglobulin G, heavy chain is mostly containing 440 amino acid, while light chain containing about 214 amino acid. Further, due to its sedimentation coefficient, it is also known as a 7S immunoglobulin. So you have to remember another name of IgG, that is 7S Ig. Further, it is made up of the heavy chain that is called as a gamma. So name is given immunoglobulin G. While light chain may be of a kappa or lambda variety, simple same that variable region of the heavy and light chain that is helping to make a enormous number of the antibody by this immunoglobulin G. Further, this immunoglobulin G is a major antibody. It constitutes about 75 to 80% of the total immunoglobulins in the circulation. So most of the immunoglobulin are belonging to this IgG class. Further, it is the antibody seen in secondary immune response. Now what is secondary immune response? So whenever the antigen or bacteria that is entering for the first time, that will lead to the primary immune response. When that same antigen or bacteria that is entering in the body for the second time, the immune response that is called a secondary immune response. And mostly in this secondary immune response, this IgG antibody is present. And due to this variable region of the immunoglobulin G, it is possible to make an enormous number of the immunoglobulin G. Further, it can pass from vascular compartment to the interstitial space. It can cross the placental barrier. It is most important barrier, placental barrier, which is in between the mother and fetus. And this crossing of placental barrier have advantage as well as disadvantage that we will look. So first, it is immunoglobulin G can protect the newborn child from the infection during the initial few days or few months of the life. So this maternal antibody, which is derived from the mother to the child, are seen in this neonatal circulation up to 2 to 4 months, and that is advantage to prevent any infection in the newborn babies. But sometimes Rh isoimmunization due to the placental crossing of immunoglobulin G is seen. So these occur particularly when mother is Rh negative and fetus is Rh positive. So when ABO system antigen are similar to both mother and fetus, but Rh negative and Rh positive, that is creating problem. So mother is Rh negative, then mostly this type of problem is created. During birth, what happen? Fetal RBC, fetal red blood cell, may enter into the maternal circulation and leading to the formation of Rh anti-Rh antibody in the mother. So basically when this child which is Rh positive and few RBC of this Rh positive that is enter in the mother circulation, and that will lead to the anti-Rh antibody generation. So during next pregnancy, this antibody which is present in mother being immunoglobulin G class, because next time it will be creating a secondary immune response, and that can enter into the fetal circulation and causing the fetal hemolysis, neonatal jaundice, and severe cases neonatal death or miscarriage can be happen. So better to avoid it for this particular condition, and what we can do that we will look. So first, passively injected anti-Rh antibody when injected within 24 hours of delivering of the first child. So if you are knowing that mother is Rh negative, fetus is Rh positive, then it should be better to give anti-Rh antibody within 24 hours of the first child's delivery. Will avoid this isoimmunization and protect future pregnancy. Further, in cows, placental crossing of immunoglobulins does not occur, but the colostrum, first day's mother's milk, contains good quantity of antibody which can cross the intestinal walls of the newborn cows. But in humans, it is generally not occurring. Human colostrum contains immunoglobulin to a lesser extent. So that is the main thing that you have to remember about Rh isoimmunization. The immunoglobulin binds with the microbes, and Fc, that is fraction crystallizable portion, can fix on the macrophage so that phagocytosis can occur. This is called a opsonization activity by the antibody. Further, antibody coating the target cell will sensitize them for killing by the lymphocyte. So, and that is called antibody dependent cell mediated cytolysis, short form, that is ADCC, antibody dependent cell mediated cytolysis. And only IgG, immunoglobulin G, is active for this ADCC mechanism. And that's all about the important part of the immunoglobulin. Further, immunoglobulin M that we will discuss later on.